Hi, yeah, thank you for inviting me. I wish I was there in person, but I'm in New York today. So yeah, although my lab is at University of Oregon, but I'm spending the spring term in New York, visiting the Flatiron Institute and NYU for a mini sabbatical. So yeah, so I did grad school in Trieste at CISA, and Damian and I were there together. And at the time, I guess I wasn't doing neuroscience; I was doing physics, and we used to.
Me neither. Me
Yeah.
neither.
So both of us switched to neuroscience. I did it much later than Damian, I guess, but I finally saw the light too. So it's really great to be here together and discussing. So I prepare a talk, but I would like you to stop and ask questions as much as you like. So it doesn't really matter if I go through the material, as long as we have a nice discussion. So please interrupt as much as you like. Hopefully that's going to be fun. And so today I'm going to talk about neural mechanisms of optimal performance. But before I start, I wanted to showcase my lab at the University of Oregon. This work is going to be, so I'm going to talk about, depending on how long we go, work that's being done by Leah Papadopoulos, who's a postdoc in my lab. She's a spectacular postdoc who's a collaborator with Damian also. so they work together when Leah was a grad student in Danny Bassett's lab. And so I will also mention, hopefully, some other work by Marav Stern, who's a postdoc in the lab, and Nico Istrate, who just graduated. And so this is all work funded by the government here, by the NIH. I also wanted to advertise that at University of Oregon, we have a very cool theory group. If you've never heard about University of Oregon, so it's in the Pacific Northwest. So we just started a neuro AI center. My other computational neuroscience colleague is James Murray, who started recently. And this neuro AI center is including systems neuroscience and neural engineering and brain-machine interfaces, including Felix Deco, Tim Gardner, Chris Neal, Dave McCormick, a lot of other people. And so today we're going to talk about a collaboration that's taking place in our University of Oregon. And so the main characters here are Leah, who did the theory part, and Daniel Halsey and Sue Zhou. So they did the experimental part. So Daniel and Sue are in David McCormick's lab. And so this is a collaboration between my lab, David McCormick's lab, Mike Weir's lab, and Santiago Jaramillo's lab. So these are experimental labs that study also auditory cortex. And so this is all unpublished, and it's based on these 5 preprints that are on the archive. So we can, I can show you more details later. Okay, so the topic of the talk is the following. So we're going to talk about optimal performance. And so I'm going to start by motivating what I mean by optimal performance from kind of popular science point of view. And then I will define optimal performance in terms of a model of behavior and decision making. I'm going to then move to study the neural correlates of optimal performance in auditory cortex of mice. And I will show you what happens what happens at the level of neural populations when we have optimal performance. Then I'm going to switch gear and move to theory, because we are theoretical neuroscientists. We want to understand the principles behind this optimal performance idea and what is the computation that's being optimized in cortex during these periods of optimal performance. So I will introduce neural network models to study these questions. And then hopefully, but I don't think I'll have time, but we'll see. I'm going to use this idea of a computational principle that we uncovered in cortical function to design a new kind of brain-inspired recurrent neural networks that can perform multitasking without any training. So this would be the AI application of this story. So what's optimal performance? So this is a very, very well-known story from more than 100 years ago. This is where what happens when you're in the flow or in the zone, and hopefully it happened to most of you. I don't know how many of you have experienced being in the flow, for example, when you do neuroscience or when you code or when you, yeah, you want to, let's take a poll. So do you want to raise your hand if you've been in the flow sometimes? I see. Anybody? Well, one person, two, let's see, two people, 3, 4. Okay, that's awesome. Okay. Okay, many of you experienced this feeling. So now you're gonna use your introspection to see when I'm gonna talk about this, if it corresponds to what you've experienced when you were in the flow. Thank you for participating to this poll. So the idea is the following, that when you're involved in complex cognitive task, your performance depends on the arousal level. So for example, when you are at low arousal or you're drowsy, kind of sleepy, your performance is terrible. For example, if you try to do some coding, right? When you're at very high arousal, which means very stressed, very anxious or scared, or running, for example, then your performance is also pretty bad, like here. But there's an intermediate level of arousal where your performance is optimal. So you perform extremely well, like in this woman who's debugging 3 screens at the same time. So there's an inverted U relationship between performance in a task and arousal level. The goal here is that we want to now define this in an operational manner so that we can study in the lab. And so this is being called, again, being in the zone or being in the flow. And so I prepared a little video clip. So now we want to study this using mouse models. And we want to draw inspiration on how to design an experiment using mice that can access this kind of full range of arousal. And so I hope it works. So one of my favorite shows is Big Bang Theory. And so this is what they do in Big Bang Theory.
Done. 33 seconds. Hey, that'll be our baseline.
You know, I don't want to tell you how to do your job, but when a mouse completes a maze, it gets a food pellet.
It also gets its brain plucked out with tweezers.
And its last meal was a food pellet? You're a monster.
Okay, so in this first part of the clip, what you see is Sheldon involved in a complex cognitive task, which is to trace this maze, like, and get from the beginning of the end of the maze, like you see here. He's performing really, really well because, you know, he's kind of in the zone. It was like very focused on the experiment. So that would be the optimal performance level that you're kind of used to when you're really doing very well. But now we want to see what happens. How can we modulate this arousal level to run an experiment on mice?
All right, next we're going to introduce an anxiety-inducing stimulus, measure your stress levels, and see how it affects your problem-solving rate.
Very well. And good luck. I'm a pretty laid-back guy.
Ready? Begin. Why'd you pop it?
I'm sorry, I was aiming for your heart.
Cool. So as you saw in this clip, then when Sheldon was getting very stressed, very, very high arousal, which is scared or annoyed or kind of anxious, it was not performing the task at all. It was not able to perform the task, right? So now we're gonna do the mouse version of this clip. So now we're gonna switch to experimenting mice. Question from Damien? Yes.
So just a question now, maybe to avoid asking you
Yeah.
later. So this is a distractor, but is also an anxiety-inducing distractor. Is there a specific difference between a generic distractor and an anxiety-inducing one?
Yeah, so what we're gonna focus on is things that modulate kind of the internal state of the animal. So that induce So a range of arousal where the animal explores all the way from kind of being very anxious, when mice are anxious, they run, to being kind of sleepy or falling asleep, actually. So it's really, there's a difference in which we're really measuring this kind of internal state that's modulated by neuromodulators, for example, norepinephrine. So it's not like a cognitive distractor, it's really like an internal state. That's yeah, that's very important for what we're gonna talk about. Thank you.
Thanks.
Yeah, so this work was inspired very much by previous studies by the McCormick lab, who worked on a mouse model of arousal before. So what they did is that they had an experiment, this is in 2015, where they had a mouse on a treadmill, and they monitored the arousal level by looking at the pupil size. So there's a long history in neuroscience and in psychology of measuring arousal level with the pupil size, such that the size of the pupil is proportional to the arousal level. For us, it would be the definition of the arousal level. That would be the size of the pupil. So this mouse was involved in a auditory detection task, a very hard auditory detection to detect a tone within a noise cloud. And so this was a go-no-go task. And what they found was that the hit rate in the go-no-go task had an inverted U proportional to the pupil of the animal, such that at low and high pupil size, the performance was very, very bad. And it was an intermediate pupil value where the performance became much, much, much better. So performance, again, had an inverted U as a function of arousal. So this was our inspiration, but we wanted to kind of get a little bit deeper into how can we characterize these performance states. And so in particular, is there like a continuous performance, or are there kind of like different strategies that the animal is deploying to solve this task or to solve tasks in general at different arousal levels? And so to do that, we designed a decision-making task. Now, it's different from what the McCormick lab used before. So this is a two alternative force choice task where the animal is sitting on a treadmill and is presented visual stimuli on a screen. And so they can, these are drifting ratings. If they are depending on the orientation, they know that they can lick left when the orientation is more vertical, or lick right when the orientation is more horizontal, and the licks are from two spouts in front of the animal. And so there's an association between a stimulus and a drop of water on the correct spout. So this is a standard task that people have been studying in neuroscience. We have an a visual version of this with the screen, and also an auditory version of this with a speaker that plays a tone cloud. And when the frequency is mostly high, then the animal has to lick left. When the frequency is mostly low, then the animal has to lick right. So this is kind of the two choices that the animal has at any trial. However, there's a third choice the animal can perform, which is not to respond at all. So this is a modification of the two alternative force choice task, where we have three choices: lick left, lick right, or not lick at all. And we'll see that's super important to study what happens in this kind of no response trials. And so in general, we can capture the full behavior of the animal using a psychometric curve. I assume you guys are familiar with the psychometric curve. So in this case, we have three choices. So there's three curves. So this is slightly different from what you usually see. So there's the probability of choosing left with a left triangle as a function of the stimulus, the probability of choosing right as a function of the stimulus. And you see they kind of, one goes down, the other goes up, because those are the correct stimuli at the left and right extreme. But there's also a third psychometric, which is the probability of not choosing, which is the one with the circle. So for every value of the stimulus, the three curves sum up to one, because that's the total probability. And so we can describe this this decision in terms of a softmax. That's called a softmax. It's this categorical decision probability. And typically, the softmax gives you the probability of one of the three choices as a function of the stimulus U that is weighted by a weight W that depends on which of the three curves you're choosing. So you have three sigmoids, basically. So this is what people usually look for when they want to characterize the behavior. But because we want to see how behavior is modulated by arousal, we wanted to test whether the animal is actually using different psychometric curves for different values of arousal. So that would be our hypothesis. And we were inspired by this kind of work, recent work by the Pillow Lab. This paper, this beautiful paper, I encourage you guys to take a look at, by Zoe Ashwood, where they studied whether when the animal was making mistakes in a 2AFC task, there was any temporal structure in how error trials occurred. So, you know, the error trials could just be occurring randomly, but what they tested was whether error trials were correlated in time. And so they introduced this idea of a GLM-HMM. So it's a hidden Markov model with generalized linear model emissions. So what's a generalized linear model? Well, a generalized linear model is a psychometric function that's a sigmoid whose output depends on the input stimulus, so also called input-output model, if you want. So what they assumed is that they tested the hypothesis that the animal is using different psychometric curves at different trials. And so then the different psychometric curves represented some internal state that the animal is experiencing and deploying a psychometric curve depending on the internal state. So you have a psychometric curve for optimal performance, a psychometric curve from right bias, meaning that any stimulus you just lick right all the time, and a psychometric curve that's very, very kind of shallow for kind of bad decision periods. And what they found is that there was a kind of a temporal dynamic. So when you deploy the same psychometric curve for several trials, and then you switch to a different psychometric curve for other several trials, so such that there's an underlying hidden Markov model or underlying Markov chain that determines the dynamics of which psychometric curve you deploy at every time point. So we took this idea and we extended this to this softmax emission, GLM. And so now in our model, we'll have the same softmax as before here, where we have three possible choices, three psychometric curves. But now the dependence of the on the stimulus in terms of these weights depend on some hidden states that we don't observe. So we have an ensemble of psychometric curves at each trial, we decide which psychometric curve set we're going to pick. And there's going to be an underlying dynamics of these hidden states that's determined by a Markov chain. So that's called the hidden Markov model with multinomial generalized linear model emission. So this is kind of the model that we use to capture behavior. So in practice, this is how it works. So if you look at the left side here, on this left side plot, what I'm showing you here, the dots are the actual decisions in each trial. So the x-axis is the full session, one example session here. And here we have the hits on top. And then on the middle, we have the errors where the animal is responding, but is responding with the wrong decision. And then at the bottom, we have the no response where the animal is not licking. That's the third choice. And you see that there's some dynamics if you just eyeball this kind of cloud of points here. And what this GLM-HMM does is that it segments all these decision-making trials to pick the intervals where different psychometric functions were deployed. And so, for example, we found that at the beginning, the animal is in a left bias psychometric curve. Then it becomes disengaged, and you see disengaged means that they don't respond at all for a lot of stimuli, and so on and so forth. And then there's long periods where the animal is performing almost perfectly with very, very good performance, but they switch back and forth. And so what we find is that there's, for this particular animal, for example, the animal uses 4 sets of psychometric curves that we call optimal. Here you see the optimal performance as almost 100% for these stimuli, and basically no withdrawal of the leak, very low withdrawal. There's a disengaged state where the animal is not responding at all, and this corresponds to these periods where the animal is really not responding at all. And then there's a left bias state where the animal is mostly responding by choosing left for any stimuli. And then there's a right bias state where the animal is mostly responding but choosing right for any stimuli. And so these strategies, we call them strategies, are long-lived, so are deployed for many trials consecutively, and then they switch abruptly in 1 or 2 trials, and so on and so forth. So this is one example of how we define, how we capture this variability in the animal behavior. And so if we take the average of these different states here, these different psychometrics, we get back to the original psychometric curve that I started from, which is the marginalized psychometric curve over all the hidden states, if you want. And so this approach is very powerful, because if we compare the prediction accuracy, how well we can predict the animal behavior in trials that we didn't use to fit the model, so we can do cross-validation to assess how well the model is predicting the animal behavior in unseen trials, we see that the prediction accuracy of the HMM is much higher than the prediction accuracy of a single psychometric curve. So this cross-validation shows that this model is actually very powerful in capturing the animal behavior. And so these are the different strategies that the animal deploys in different parts of the session. And we characterize then optimal performance in terms of this optimal state, where the animal is these intervals where the animal is performing at basically almost 100% of the responding to the stimuli. And then there's several suboptimal states. Some of the suboptimal states are where the animal is still responding but making mistakes, the bias state. And then there's states where the animal is not responding at all, these disengaged states. So this is like a characterization of what's the spectrum of strategies that the animal can deploy for decision making. So now the question is, can we predict what are the characteristics, what are the features of these different performance states? And so what we did is, like, we monitored the animal's face movements and like running with many cameras. And so what I'm showing you here is just an example of the animal moving whiskers, and you can see the pupil growing and slowing down. And here, this is a moment where the animal is switching from the optimal to the suboptimal performance state. And what we want to do is we want to correlate these different strategies, these hidden states of performance, with this kind of facial expressions of the animal. And what we found is that if we plot now the probability of each of these states, for example, in gray here, you see the probability that you are in the optimal performance state as a function of the pupil size. We find that you are in almost certain to be in the optimal state when you are at intermediate pupil size. This is the pupil range, and you're never in the optimal state at low and high pupil. So this is this kind of incarnation of this inverted U-shaped curve that I told you before in terms of strategies that the animal is deploying. And on the other hand, the flip side of this is that the disengaged states, where the animal is not performing, has the opposite shape, right? So it's very, very likely that low and high arousal states, and it's never in the disengaged state when you are at intermediate pupil size, this medium pupil size. And the other states are kind of have a uniform distribution. These kind of states where the animal responds, but they make some mistakes, these bias states, they're kind of uniformly distributed. So this is the inverted U-shaped version of the Yerkes-Dodson law, if you want, for a 2AFC task of this animal. And we also find, okay, a long story short, but there's a lot more interesting stuff going on, meaning that we can also predict this inverted U of the optimum performance as function of the facial movements and the running speed. So the actual uninstructed movements also predict this optimal performance state. So this is related then. Yes, question.
We have two questions, one from Damian, that he's asking if the number of case states imposed or self-selected by the fitting algorithm.
Yeah, thank you. So I was very quick to sweep this under the rug. So you have to do model selection now. Whenever you have a hidden Markov model, then the question is, how do you pick the number of states? Thank you for the great question. Then we spend a lot of time thinking about that in the lab. So if you fit many different hidden Markov models to the same data, as you increase the number of states here, this is in this plot, on the x-axis, you see the increasing number of states in the model. On the y-axis, you see the cross-validated log likelihood of the holdouts. Again, you train the model on half of the data, and then you calculate the likelihood that that model explains the other half of the data. You see that it increases until you find an optimal number of states, which is in this case for this particular animal. So we did it animal by animal. And even though we fit a different model for each animal, we find the same states in all animals. Basically, yeah, they're very congruent across animals, and there's a whole story there. But so if you want to see the details, they are in this preprint that we just put on the archive with all the kind of, all the statistics, all the story there. So yeah, other question?
Yeah, there is another question of Wesley Clausen that he say that he missed it. That are you doing EFIs at the same time of this task?
Yeah, so thanks for the question. So now we're gonna switch gear to answer the question. Yeah, so that's the second part of the talk. So what I showed you so far is the behavior, and now we wanna kind of come up with a hypothesis as to what's the neural mechanism that drives this optimal performance. So what's the neural correlate? So let's brainstorm a little bit. So the first hypothesis is that this optimal performance, these kind of different values of performance, arise at the early stages of information processing in the brain when the information comes into the brain. So this is, for example, an auditory task. Then this is already modulated at the level of auditory cortex. So, the first stage of processing in cortex for auditory information coming from your ears. So that's the first hypothesis. The second hypothesis is that actually maybe these arousal-related modulations of performance come along like at the late stage of processing, like, when the information turns from sensory to motor. So prefrontal areas like, I don't know, secondary motor cortex, or even posterior parietal, something in the middle, or prefrontal cortex. So something kind of some cognitive area, right? So these are two potential ways that these different performance could arise in the brain. And so what we're gonna do is we're gonna test hypothesis 1, because if hypothesis 1 is correct, then 2 could also be correct. And so it's kind of like the first hypothesis to check, right? So if you just test hypothesis 2, you find modulation in the frontal cortex. You don't know if there's also modulation in the sensory cortex. But if you test hypothesis 1, you're sure that that's the first stage where this modulation could occur. And then hypothesis 2 could still be valid as well. But we will test it later, right? So we're going to start from testing hypothesis 1, which is whether arousal modulations already occur at the level of primary sensory cortex, in this case, auditory cortex. And so to do that, we have to disassociate the sensory effects of arousal from the cognitive decision-making effects of arousal. And so in order to do that, we designed a different task in which now the animal is just being presented auditory stimuli passively, and they don't perform any decision, right? So we remove the decision-making part of the task, and we only test whether the information is encoded in different ways depending on the arousal level in sensory cortex. So this is what we're gonna do now. So we have the same thing like an animal on a ball with videos. So we monitor the animal's face and pupil and so on, and we just present passively some auditory stimuli to the animal. And so we record neural activity from neuropixels. So we have like a lot of neurons. So this is a raster plot like a few seconds with 60 neurons, and every bar is where we present an auditory stimulus. And we do this for 2 hours. And then we interleave with periods of spontaneous activity and so on. So we have a lot of data to play with. And then we have the running speed of the animal and the pupil size. And now we're going to make sense of all of this electrophysiology together. So what are we going to do? Well, we're going to estimate the information about sensory stimuli that's encoded in cortical activity using a linear classification analysis. This is like standard machine learning. So how does that work? So we take, these are the spike responses of the population of simultaneously recorded neurons in response to one tone, like a yellow tone, for example, here. If you're synesthetic, you get a yellow tone. And we compare that to the response to a purple tone. And what we do is we just calculate the spike count of all these neurons. And this is like a 60-dimensional vector, right? So that's 1 point in 60-dimensional space. And I'm showing you here a cartoon of a 3-dimensional space with 3 cells, but imagine this is 60-dimensional. And so every time we present a yellow tone, we have a data point here. Every time we present a purple tone, we have a purple point here. And then we look for a hyperplane that separates yellows from purples. So that's a linear classifier. That's a standard linear classifier, assuming that there's a downstream neuron that's reading out the information from auditory cortex, if you want. And then using this, we can construct a cross-validated estimation of how much information about the sensory stimuli, the colors, right, are present in the spike responses of the population. And crucially, we're going to do this conditioning on the pupil size. So we're going to run a classification analysis at medium pupil like this, and then another classification analysis at large pupil. And so we'll do it pupil by pupil quantile bins. And what we found, long story short, is that the performance of the classifier as a function of the pupil size has a really beautiful inverted U-shaped curve. So what I'm showing you here is all the animals. Every line is an animal. And we found this not only at the single animal level, but even at the single stimulus level. So every frequency for every animal has this really kind of beautiful inverted U-shaped curve. And so this is the neural correlate, if you want, of the Yerkes-Dodson law that suggests that arousal is modulating the way we encode sensory information already at the first stage of cortical computation, which is primary auditory cortex. So this confirms the hypothesis one, if you want, that arousal modulates sensory processing. Yes, question?
Yeah, so maybe it's a question for the end of the talk and you're going to answer, but here you don't know yet whether it's a direct arousal effect on the sensory region or whether it's a top-down effect on sensory region, which is modulated by arousal.
Yeah, so that's a good question. So, well, in fact, this question is the origin of our whole collaboration and all the grants we write. So our goal here in general is to be able to tell apart these two different mechanisms. So typically, arousal is related to this kind of neuromodulatory signaling, which is some kind of a blanket broadcast signal that's originating in some midbrain nuclei like locus coeruleus or like, I don't know much anatomy, but they told me there's this kind of nuclei somewhere deep in the brain that broadcast this kind of neuromodulatory signals like norepinephrine level, acetylcholine level that modulate all the cortical areas at once. So it's kind of like an unspecific signal that just really measures this internal global brain state. On the other hand, there could be some top-down projections from frontal cortex that could be related to what people call attention, feature attention, spatial attention, that kind of stuff. There would be a cortical connectivity that modulates sensory cortex. So people know that that's the case. So these are different projections that modulate sensory cortex. So these two mechanisms are always present and occur together. And so I don't have anything to say about the cortical kind of attentional mechanism for now, because we're running the experiment right now with pair recordings from N2 and A1. So, but that would be the second part of this. For today, I'm not going to talk about it. I'm only going to talk about this global modulation by brain state. And then the super interesting part for me is to understand the interaction between the two, right? Because that's the important thing for our cognitive abilities. So yeah, so I'd love to talk more about that. It's like, that's the whole research project, basically, of this collaboration. So for today, it's really only this kind of global internal state. And these results suggest that this occurs already at the level of auditory cortex. And so, okay, so now...
quick question
Yes,
before you go to the next part. Do you still have the same kind of the mouse is running around at the same time while the pupil is being dilated? Like, do you still have that comorbidity, that confounding factor that the mouse is still running?
Oh yeah, so that's a great question. So what we find is that when the mouse is running, that's always in the right-hand side tail of the arousal range. So when the mouse is running, it's mostly at very large pupil size. So there's a strong correlation between high arousal state and running and very large whisker deflections. So basically the animal dilates the pupils and it starts to run and move like crazy. So this is what mice do. So if you control for that, you can remove the running trials and only looking at stationary trials with large pupil, and the effect is identical. So it doesn't rely on the running. In fact, even though they're very correlated, they're not the same, because there's a lot of kind of resting trials where the pupil is kind of getting all the way to the large arousal, to the large size. So that's kind of a super interesting question. Then can we kind of separate the effects of running from the effects of large arousal? So that we're also looking at, it's kind of like cool. Yeah, so presumably running induces some different projections from the one strictly related to the pupil. And so although the effects are the same, but the origin should be different. So
it would be interesting to sort of manipulate it and see if there's some sort of like dynamics in response to the running, right? Like you could force the mouse to run faster or slower, and depending on that, seeing if you can like maintain a certain performance, possibly.
Yeah, very much.
Super interesting.
Those are all very, very good questions. In fact, even though, you know, running is kind of generic, can you do something better? Can we actually study the effects of different classes of movements on this coding? For example, it could be running, but it could also be grooming, or it could be the animals move their face all the time and make all these kind of funny faces. Do they have all the same effect, or are there specific effects on neural representations depending on this kind of different movement states that the animal are engaged in? So we're also studying this right now. So it's a much more complicated.
Super interesting.
But and then the penultimate one is what happens when they're all freely moving rather than head fixed, because I think that might change some of the movement dynamics that you were just describing to make it one more complicated.
It's a very, very good question. So, so that's exactly what we're doing now. So Leah now has been analyzing a different experiment where the mouse was in a cage, but was just free to run around, but with a crown on top that was measuring with the camera the pupil size. And with other multiple cameras, we can, you know, kind of track the animal behavior. And at the same time, we can play tones to the animal with the headphones. And so we can test whether head fixed and freely moving have different signatures, which is probably the case, because freely moving, the internal state of the animal when they're freely moving is completely different from when they're head fixed. So so Leah just showed me yesterday a few plots with an inverted U is freely moving, but it's a much more complicated story there. But definitely that's what we're looking at right now. Thank you for these questions. Yeah, so now let's switch gear again. Now we put our theory hats on, and now we ask the following question. So what is the computation that's going on in primary auditory cortex that results into this modulation, this inverted U? So now we're going to try to model this. And how do we model that? Well, we model it in terms of recurrent neural networks. So I hope these things are familiar from, at least for Damian's lab. So we use networks of excitatory and inhibitory neurons. These are recurrently connected in balance state. So these are spiking networks. So neurons like link integrated and fire neurons. And so this is kind of a cartoon of what that's like. And so the first question is, how are we going to model arousal in this EI recurrent neural network? And so because I already mentioned that arousal is related to this kind of neuromodulatory projections that people know, that are projections from midbrains through axons that project to primary auditory cortex, we're going to model arousal as a baseline input, an external input to this recurrent neural network. That's the local circuit in auditory cortex. And so this is arousal modulation is a change in the baseline input, the synaptic input to these neurons in auditory cortex. And so now there's a whole zoo of possibilities in these EI networks. So arousal could be projecting to excitatory neurons or to inhibitory neurons. And moreover, it could change the mean of the input, or it could change the variance of the input. So there's like 4 cases here that we can map. And then we're also going to explore alternative models in terms of the circuit architecture. So, for example, you have an EI network that could be in a vanilla balance state without any structure. We call it unstructured network. Or you can start to put assembly structures in this network or neural clusters of EI groups of neurons that are more strongly connected, and you have a bunch of them. So we're going to compare unstructured network with clustered architecture. So this is kind of the space of models that we explore to capture these effects of arousal. So let's talk a little bit about this baseline first. So we want to figure out which of these 4 modulations represents arousal. So for example, I'm showing you just an example here. So we have arousal as an increase in the mean input to all excitatory neurons. This is delta, a change in the mean to all excitatory neurons here. Or arousal alternatively could be a change in the variance across neurons. So a low arousal, all neurons get the same input, but a high arousal, we change the value of the mean value of the input to be larger for some neurons and smaller for some other neurons. So we increase the variance across neurons, and so on and so forth for all the other cases. So in the models, if you play with this, then we want to compare the effect on firing rates of neurons in response to this change of arousal here. And if you increase the mean to excitatory neurons, all neurons go up. 100% of the cells increase their firing rates, right? That's obvious. But if you change the variance, you find that half of the neurons go up and half of the neurons go down, right? So these are 2 signatures of these different modulations at the level of population responses to arousal. And then we go back to the data and we look at, so how do neurons in the data respond to arousal during spontaneous activity when there's no stimuli, right? So this is what we did. And we find that half of the neurons go up, half of the neurons go down. So arousal is compatible with this increase in the variance across excitatory neurons in their baseline input. So this is going to be our model of arousal.
But quick question. So you're just the arousal is only projecting to the excitatory parts or also to the inhibitory?
Only the excitatory ones, yes.
Okay.
Although you can, then we tried all of them and this was the only one that worked. So basically the other ones were not compatible. But there's a whole slew of stories here. If you're interested in all the other ones, I wrote a paper with my grad student, David Wyrick, where we kind of classify all the possible perturbation effects. It's kind of a complicated story, but for now, the only one that works is that this increase in the variance of the input. Okay, and then we're gonna compare these different network architectures. So in an unstructured network, so this is a synaptic weight matrix in this EI unstructured network. People know that these networks generate just asynchronous irregular activity. So these are just like, you don't see any structure in these spiked rasters of all the neurons here. But if the network is clustered, so if you have this kind of Hebbian assemblies in the network with preferential strength and synaptic connectivity between neurons within the assembly, then the network becomes metastable. So during spontaneous activity, in the absence of any input, you find that these clusters switch on and off their activity at random times. So different clusters activate and inactivate randomly. So you have this metastable, interesting metastable dynamics with a long time scale. that emerges from the network dynamics. So we're going to compare the effects on these two different architectures. And so how do we model stimuli now? So now we model arousal. Now we want to model auditory stimuli. So we just say that every group of neurons, a random subset of neurons, is selective to one of the stimuli. And then another random subset of neurons is selective to another stimulus, and so on and so forth. And the selectivities are overlapping. So every neuron has a certain 50% chance to be selected to any stimuli, basically. So there's random mixed selectivity. And now we put all together. So we perform the experiment in the neural network, right? So we have stimuli, auditory stimuli. Then we have this arousal modulation. We put everything together. And what we want to do is we want to now perform the same classification analysis that we did in auditory cortex at different levels of arousal. We want to predict how much information about the stimuli is present in the population spike counts, just the same exact thing that we did in the mouse. And we want to compare the effect on unstructured network and cluster network. So what I'm going to show you now is, as a function of this variance increase, which is our model of arousal, what the performance of this classifier is like. And what we find is that in the structured network, increasing the variance just decreases performance. But in the cluster network, yes, question.
For clarity, so in the unstructured case, the neurons still have a selectivity. It's just salt and pepper, not related to connectivity.
Exactly. Yeah, it's salt and pepper random mixed selectivity to all the stimuli. In both cases. So, except in these cases we have Hebbian assemblies. In this case, there's no assemblies, but it's the same selectivities. Does that make sense? Oh, hello. I think did Damien
he's either deep in thought or he disconnected.
Maybe he fell asleep, his eyes open. Okay, so what we find is an inverted U-shaped curve between arousal, this variance modulation, and the performance of this classifier that shows that at intermediate level of arousal here, the network is encoding the sensory stimuli the best. And this encoding is worse at both low and high arousal levels. So this basically shows a hypothesis for what kind of computation is going on in auditory cortex that's realizing this inverted U-shaped curve. And so this is the model, this is the data, it's kind of very similar. All right, so but now I still haven't talked about what's the computation. So we show that the model is actually reproducing the neural data, right? The mouse data. But now we want to get a little bit of insight into what computation is actually modulated. So how is it that we get this inverted U-shaped curve? So to get to that, we need to switch gear a little bit and do some theory now. So these are like network simulations. Now we want to do theory. So how do we do theory? We use what's called mean field theory to solve the network attractor structure. And so the idea is that you can use mean field theory to calculate the stable fixed points of this network with Hebbian assemblies. It's a bit complicated, but you can use this kind of fixed point equations that give you the firing rate nu in each cluster as a transfer function of depending on the input current and input variance, which are also function of the firing rates. And so there's a self-consistent solution to the stable fixed points of this network. And so in particular, there's a way to extract an energy potential, an approximate landscape for this network where attractors are represented as these potential wells calculated from the mean field theory by integrating the mean field theory equation. And so different potential wells are the locations of these attractors. And then these metastable dynamics, the switching dynamics that I showed you in the simulations, represent switching from one well to the other. And the transitions are driven by noise, so that the switching probability depends on the height of the barrier separating the attractor. So the larger the barrier, the slower the switching. And as you shrink the barrier, we switch faster. So that's kind of the picture that you get from mean field theory. And so what we found now is that we want to see how these barriers are modulated by arousal, right? So arousal is this increase in the variance. And what we find is that at low arousal, there's high barriers, but as we increase arousal, the barrier size shrinks up to the kind of a point where there's a phase transition from this multi-stable phase to a phase where the barriers are just flattened, the whole landscape is flattened, and you're just back to one single attractor. So arousal drives a phase transition from a multi-attractor state here to a single attractor state here to the right. So this is kind of the calculation that's going on in the network. And then now we can plot the effect of arousal on the dynamics here and to look at how the performance does. So at low arousal, performance is bad. And that corresponds to slow switching. These are like, we call it stiff clusters. And the idea is the following, that you have this ongoing metastable dynamics of slow switching. And when a stimulus comes in, the network has to jump into the correct attractor from this other, whatever random attractor it was on, right? And so it takes quite some time for the network to switch if the clusters are stiff, if the barriers are high. But when we increase arousal shrinks the barrier between the attractors, such that now when we present the stimulus, it's very fast to switch into the correct coding attractor, if you want, and that's at the top optimal performance, so that we have flexible clusters now. But if we increase arousal too much, you completely destroy this attractor structure, and so coding like becomes bad again, because you don't have this kind of big separation from coding to non-coding attractors anymore. So there's an inverted U-shaped curve then between performance and arousal that's determined by how arousal is modulating the switching dynamics, such that just before the phase transition, there's this optimal flexibility of these clusters that give you optimal encoding when you present a brief stimulus. So this is the calculation that's going on in the network. So we propose this as a computational principle, if you want, of how to flexibly modulate performance in a simple encoding task like this as a function of arousal. And so this relies on this cluster architecture, on these Hebbian assemblies that kind of has this slow switching dynamics. So arousal induces a phase transition in cortical activity. That's our claim. So now there's one clear prediction of the model that we have not talked about. So we only talked about the effect of arousal on performance, but arousal here modulates neural variability very much. So here you have slow switching at low arousal, fast switching at intermediate arousal, and no switching at all at high arousal, like asynchronous dynamics. So the prediction of the model is that arousal should monotonically decrease the neural variability. So here we have very high variability, right? Neurons are very correlated and they switch. And here there's basically no variabilities like Poisson, like sub-Poisson variability. So there's a monotonic decrease of variability with arousal. How do we measure variability? Well, the simplest thing is the Fano factor of single neurons. That's the variance of the rate divided by the mean of the rate, right? So this is very high here and very low here. And in the model, you find, in fact, that the Fano factor is kind of monotonically decreasing as a function of arousal. So that's pretty obvious from the raster plots. So now what we did is like, so we went back to the data, and now we looked at what happens in the mouse data as a function of arousal, what the Fano factor does. So we looked at periods of spontaneous activity now. Sorry, when there was no stimulus delivered, right, in the same experiment. We have these long periods that we put in the experiment, and we tested whether the Fano factor was modulated by the pupil size. And what we found, in fact, is a very strong modulation of the Fano factor as a function of the pupil size in all animals, which is monotonically decreasing as predicted by the model. So let me wrap up this computational part. So we found then in the task, this inverted U-shaped curve relating the optimal performance state with the pupil size. In the auditory cortex, we found that the encoding of these auditory stimuli as this inverted U-shape in terms of as a function of the pupil. In the cluster network model, where we have the switching dynamics, we found that the encoding of auditory stimuli has this inverted U-shaped curve now as a function of variance, this variance in the input currents that represents arousal. And using the theory, we explain this in terms of this phase transition from multi-stable to single attractor phase. And so now I want to step back a little bit. And okay, so I just showed you this relationship between arousal and modulation in auditory cortex with Leah. So in previous papers, we showed that this is also capturing the effect of running on visual cortex. And this is this other paper with David Wyrick, student in the lab. This is running, the effects of running in visual cortex can also be explained in terms of this variance modulations and in modulations of cluster network or this metastable dynamics. In previous work, I showed that when I was a postdoc, that these variance modulations explain the effect of expectations on gustatory cortex. So this is taste encoding, right? So auditory, visual, and gustatory. And in this kind of recent paper with Stefano Recanatesi, we showed that the same kind of models of metastable dynamics explain the timing of self-initiated actions with the same mechanism in premotor areas, secondary motor cortex. So we would like to propose that this kind of modulations of metastable dynamics are like a canonical way for cortex to implement this flexibility of information encoding or motor dynamics in motor areas. So it's kind of a canonical cortical motif that we found across different sensory modalities, tasks, and different cortical areas. So okay, all right. So that's what we found in physiology, if you want, in electrophysiology data. And this is kind of a biologically plausible model that I showed you so far with EI spiking network. And so what I wanted to now in the last how much time do I have?
It's fine. We made you a lot of questions, so you can still go on 10 minutes.
I can go.
5,
okay,
10 minutes.
So how long? Sorry, say it again.
5, 10 as you want.
5, okay, great.
It's informal, so don't worry.
Awesome. So I have time to talk about the last part, which is very exciting. So now I wanted to kind of draw some ideas from this story and try to look at the machine learning approach of how to use arousal to do computation. So I showed you here that quenched disorder. So from a physics point of view, we call this changing input variance as quenched disorder in the inputs to this local circuit. Induces a phase transition, right? So what we did is we modeled the baseline B to each neuron I as drawn from a Gaussian distribution with zero mean and variance sigma. So that's kind of how we in practice, how we implemented this baseline, this quenched disorder. And what I want to do now is I want to take a step back and have a simplified model to study in general the effect of these input baseline modulations from a machine learning point of view or from a reservoir computing point of view. So what do we do now? So now we take a much more simplified model, which is a recurrent neural network in discrete time. So this is a recurrent neural network of units xi with random couplings jij. This is the standard reservoir computing framework, if you want. So random couplings, now completely random. We take positive activation functions like a sigmoids, as people do. And now we take the new input. The new ingredient is that every neuron gets an input bi, like a constant input that's drawn from a Gaussian distribution with mean mu and variance sigma. So this is kind of abstract version of what I did before. Right? Before I had a biologically plausible model with spikes and EI balance, blah, 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 and clusters. Now forget about that. We take like a spherical cow approach to this, which is just a recurrent neural network with random couplings in discrete time. And we want to see what's the effect of changing the mean and variance of the inputs on this network dynamics. So to do that, long story short, you can solve analytically the phase structure of these networks using this MSR path integral or what's called dynamic mean field theory. and we can capture the phase diagram of the network in terms of this self-consistent equation of the mean and variance of the activity now, not the input, but the actual activity of the network. And you can capture the dynamics in terms of the largest Lyapunov exponent of the network that you can actually calculate analytically using dynamic mean field theory. So everything is under theoretical control if you want. And this is what we found. So now we want to calculate the phases or the Lyapunov exponent, if you want, of the network as a function of the mean and the variance of these input currents, right? So map out the whole phase space. And this is the phase space that we found. So we found some phases where the network is in a fixed point. That's the blue region. So it's a fixed point. So you initialize the network and it reaches just a fixed point. Other phases like this, in this phase, the network is chaotic. These phases are well known. So the network is just having this chaotic dynamic. So positive Lyapunov exponent. But we found some super cool new phases where for some values of mu and sigma, the network has bistable phases, meaning that for the exact same value of the parameters, if you initialize the network in some initial condition, you get to a fixed point. If you initialize the network in a different initial condition, you get to chaos. So there's different basins of attractions of initial conditions such that you end up in one of two phases, bistable phases. How does that show up? So in the Lyapunov exponent point of view, so the Lyapunov exponent here as a function of sigma, as we move up this line, now we move up a line where we fix the input mean and we just increase the variance. We move from a fixed point phase, that's a negative Lyapunov exponent here, into a bistable phase where the Lyapunov exponent bifurcates here at the phase transition, and you can keep going on the fixed point here, but there's a second phase appearing for the same value of the parameters where you have there a second Lyapunov exponent that's positive, and that's a chaotic phase, until when you cross this phase boundary, the first phase dies and the chaotic phase survives. And so you can map out using Lyapunov exponent this kind of cool structure of bistable phases. And so, for example, we have a lot of cool new phenomena, which is this is disorder-induced chaos, right? So people thought before that noise or disorder would suppress chaos, but here we find that actually it's very complicated, and noise can also induce chaos in some regimes. If we zoom in this tricritical phase, we find some more interesting phases here around this tricritical point. And so this is a phase where we have two different fixed points. So if you start from some initial condition, you end up in one fixed point. If you start from another initial condition, you end up in another fixed point. So now the question is, can we use this phase structure to perform tasks or to perform computations without any training? So, you know, so far what people typically do in the field is that they train RNNs using gradient descent to learn tasks, right? That takes like, I don't know, 100,000 iterations, blah, blah, blah. So can we do that without any training? This is a random neural network, an open reservoir, what people studied, I don't know, 30 years ago. And so how do we define a task here? So this is how we define a task. So we have, this is the time course of a stimulus. So let's say we start with 0 stimuli. At some point, we turn on a stimulus a little bit for some neurons in the network, for a little interval, and then we turn it off. That we call the sample period. Then we have a delay period with no stimuli, and then the network has to report a decision at a certain time after the delay. That's one trial. So that's what we usually do in the experiment. What we did with the mice, we do the same with the network. And then you add another trial with a different stimulus, sample, delay, and decision. So this is called a delay decision-making task. And so we can set up a stimulation protocol like that. And then we can read out the network activity and decide whether the network makes this one decision or another decision. And so this is how it works. So this is the network activity during spontaneous periods here. Then we stimulate the network here. And so the network, is kind of responding to the stimulus. Then we withdraw the stimulus here. And the network during the delay period is reaching some particular configuration, one of the bistable phases. And then we read out the decision here using our linear readout, and we determine whether the network has correctly classified or incorrectly classified the stimulus that was presented here. And then we do it again with another stimulus and so on and so forth. So in each bistable phase, the network is performing a different task. The idea is that you have a bistable phase, so you can have two different responses, two different states of the network that correspond to the responses to one of two stimulus classes. And so, for example, in this bistable phase, that's the fixed point chaotic mixed phase. One stimulus drives the network to the fixed point phase. That's this one, right? So you initialize the network here. You present a stimulus. This is the network is moved to this point when you present the stimulus. Then you withdraw the stimulus. The network goes back to the bistable phase, but it goes back to one of the two branches of the bistable phase. In this case, the fixed point branch. And you see it here, the network reaches a fixed point. But starting from here, I present a second stimulus. It brings the network here. I withdraw the stimulus. During the delay period, the network goes back to the delay phase, to the bistable phase. But now it's on the other branch, the chaotic branch. And so it reaches this chaotic value of the bistable phase. And so the network is performing a go-no-go task. So a go task where the network is generating a chaotic activity in response to some stimuli, and a no-go task where the network is just still, they're not performing any movements, if you want, in response to the stimuli. So that's one task the network can perform. And again, the network is performing this task using this kind of neural hysteresis. In this Lyapunov exponent plane, I showed you, you start from here, you present a stimulus, then you go back, but you go here. But if you present another stimulus here, you go back on the other branch. So you can control which network the branch will be in the bistable phase by presenting stimuli. But the network now can perform multiple tasks for the same value of the J's of the recurrent connectivity, if you want, just by changing the input mu and sigma. So for one value of mu and sigma, the network has this bistable phase. But for a slightly different value of mu and sigma, the network is in a different bistable phase, the double fixed point phase, for example. So just by changing the input mean and variance, I can switch between different tasks that the network can perform without any training. Again, because the couplings are random. The JAJs are just random. We haven't trained anything, right? And so now we can write down a cool stimulation protocol where we pick one value of mu and sigma that corresponds to one task, this phase, for example. And in this task, we present a stimulus, another stimulus. So we perform the task just like the animal. Now we switch then at some point to a different task. That's a different value of mu and sigma. That's going to be here. And then in that task, we present stimuli and get the network response, blah, blah, blah. And so we can switch between different tasks, do multitasking, just by changing the mean and variance of the input to the network without any training again. And the network performs with 100% performance without any training. This is the readouts of the network. This is the activity, and so on and so forth. So we're very excited about this because, you know, this is a way to do multitasking that doesn't require any training. That's just an open reservoir. And this suggests maybe that this kind of phase transitions that we found here and that we propose were implemented by arousal in auditory cortex are maybe a general way that cortex is implementing this flexibility, switching between multiple regimes or multiple tasks, just by changing the mean and variance of the input currents to one particular brain area. So coming from some other brain area, which could be either prefrontal cortex, switching tasks, or midbrain areas, just like changing the global state of the animal, if you want, to achieve flexibility in terms of information encoding, or task switching in more general cognitive scenarios. So let me wrap this up. So we have inverted U in behavior again that we explained using this spiking network EI balance in terms of this phase transition of switching dynamics here. I showed you that that can be understood as a modulation of arousal that drives a phase transition in the landscape, energy landscape of the network, if you want. And then more generally, in a machine learning and reservoir point of view, I showed you that the same modulation here can achieve this multitasking where we have ergodicity breaking, where we have these bistable phases in a machine learning point of view to achieve multitasking. Okay, so that's the end. So this is the people who did the work. So the first part, this was Leah, Daniel, and Sue, who did the experiments and the theory for the animal. The animal result on arousal, and for the last part on the phase transitions in machine learning, this is a collaboration with Shu Nogawa and Francesco Fumarola, Eriken. So this is all unpublished again. And yeah, if you're interested, these are some of the preprints. And if you like this work, I'm looking for a postdoc, so let me know if you're interested, or if you're applying for postdocs to do something in this ballpark. So yeah, I can stop here.